Hello students, my name is Jatin Dewan and let's get started with principles of inheritance and variation. So, what is genetics? Genetics deals with the inheritance as well as variation of characters from parents to the offsprings, that is from one generation to the other. Inheritance is the process by which characters are passed on from parent to the progeny, that is from parent to the child. Variation is the degree by which progeny differs from their parents. Now, the first person who invented the laws is Gregor Mendel. Mendel's Law of Inheritance states that Gregor Mendel conducted hybridization experiments on garden peas for 7 years. His experiments were successful due to large sampling size, which gave greater credibility to the data he collected. He conducted artificial pollination, cross-pollination experiments following on the characteristics he studied, and we shall be discussing about them later on. A true breeding line is one that, having undergone continuous self-pollination for several generations, Mendel selected 14 true breeding peas plant varieties as pairs which were similar except for one character with contrasting traits. True breeds were selected by Mendel. He said he selected stems to be tall and short, flower color violet and white, flower position axial and terminal, the pod shape inflated and wrinkled, the colors yellow and green, the seed shape round and wrinkled. So, the general terms we shall be discussing in this chapter are: genes are the unit of inheritance, alleles are the part of genes which code the pair of contrasting color traits, genotypes is the genetic constituent, then phenotype is the physical appearance. So, genotype is the genetic constituent, phenotype is the physical appearance, that is tall or dwarf. Then homozygous is one of the properties in which allelic pair of genes are identical, that is capital T and lowercase t's. Heterozygous is one in which allelic pair of genes are different, that is a capital T and a small t. Dominant is the character which expresses itself. So in here, we can't discuss which is dominant or recessive, but if we had three capital T's and a small t, the dominant one would be capital T, wherein in the recessive one would be the small t, which is hidden behind the dominant character. Punnett square is the diagrammatic representation of crosses and these respective characteristics. So this is a monohybrid cross wherein a tall plant is made to do a cross with small, and then later on forms 3:1. We say 3:1 on the basis of phenotype. The phenotypic ratio is 3:1 as there are three capital A's. And then dihybrid is between two kind of seeds. One was round green and the other was wrinkled yellow, or vice versa. And they are taken to be like this, and the phenotypic ratio becomes 9:3:3:1. Now let's have a look of the principles of inheritance. The first principle we will study is law of dominance. This law states that the expression of only one dominant character, please note, only one dominant character in a monohybrid cross is the F1, that is the first generation expression, and expressions of both in F2. So the law of dominance in short states that characters are controlled by discrete units called factors. Factors occur in pairs. In a dissimilar pair of factors, one member of the pair dominates the other recessive. So if you see this pair here, the capital T is dominant over the smaller one. The second one is law of segregation. Law of segregation states that allelic pairs separate or segregate during gamete formation and randomly unite at fertilization. So they say that they get out with one another, they go, have a round, and come back and unite with one another again. The alleles do not show any blending that both the characters are recovered as such in the F2 generation. Though one of these is not seen in the first one. If you see here, so we couldn't see this here, but then we can see in the, I cannot see here clearly, but we can see here. So the parents contain two alleles during gamete formation. The factors or alleles of a pair segregate or separate from one another. The homozygous parent produces all gametes that are similar, that is contain same type of allele, wherein heterozygous, which are like different parents. So here, if we see, these are hetero, these are homo here. Okay, so they produce two kinds of gametes, each having one allele with equal proportion. The law of incomplete dominance, or we can just simply say incomplete dominance. When a cross between two pure breed is done for one contrasting character, the F1 breed phenotype does not resemble either. If you see here, it does not resemble either of the two parents, and was in between the two parents is called incomplete dominance. Inheritance of a flower color, if you see here, is a good example. F2 generation is here. So there's a red plant and there is a white plant, but it gives out you the pink plant because the dominant character is given out by the red one. Then there is codominance. In codominance, it states that when a cross is made between two different or dominant characters, both the characters express itself in the F1 generation. Example, ABO blood grouping. ABO blood grouping is the best example to explain codominance. In this, the plasma membrane of RBC has sugar polymers which are known as antigens, which protrude from its surface in kind of sugar, is controlled by gene I. So if you see here, this is A, this is B, this is AB, and this is O. So these are your protruding antigens, the polymers. So the gene I has three alleles, IA, IO, IB. So IA and IB are completely dominant. IA and IB are completely dominant over I. And then IA and IB present together, they both express their own types of sugar. This is because of codominance. And then hence red blood cells have both A and B type of sugar. The example of ABO blood grouping produces a good example of multiple alleles. There are many more than two, that is 3 alleles governing the same character. Law of independent assortment. If we get on to the law of independent assortment, this is the inheritance of two genes. Law of independent assortment shall be defined as when two characters, that is dihybrid, are combined in a hybrid, segregation of one pair of traits is independent of other pair of traits. So that means one does not depend on the other for survival, it is on its own. Chromosomal theory in theory of inheritance. So in 1930, three scientists named Rice, Correns, Vaughan discovered chromosomes. Walter Sutton and Theodore Boveri noted that the behavior of chromosomes was parallel to the behavior of genes, which explained the Mendel's Law. According to Sutton and Boveri, the pairing and segregation of a pair of chromosomes would lead to the separation of a pair of factors they carried. So they combined their chromosomal theory with the Mendel's principles, thus forming the chromosomal theory of inheritance. This theory was experimentally verified by T.H. Morgan using the fruit flies, that is Drosophila melanogaster. Now, linkage and recombination. According to Mendel's law of independent assortment, the genes present on two different chromosomes, they assort independently, but if they are present on the same chromosome, they may or may not show the same character. The tendency of some genes to inherit together in process of inheritance is called linkage. All genes which cannot inherit together from one generation to the other forms a linkage group. Groups in human females and 24 in males and Drosophila has 4. Crossing over is a process that produces new combination of genes by interchanging of segments between non-sister chromatids of homologous chromosomes which occur during the tetrad stage of pectination of prophase 1 of meiosis 1. When two genes are located very close to each other, there is hardly any crossing over that can be detected, but if the distance is very large, crossing over can be detected. Only crossovers happening in this small region can produce AB or AB chromosomes. Recombinant chromosomes do form but did not form very often. Sex determination. Sex determination can be done by sex chromosomes used to check the sex of an individual in unisex organisms. The normal chromosome other than sex chromosomes are called autosomes. Sex chromosome can be homomorphic, that is similar XX, or heteromorphic, that is XY, which is present in the male sperm. Homomorphic chromosomes are present in homogametic organisms, that is male birds, human female. And chromosomes are present in heterogrammatic organisms, that is female bird and human male. Out of 23 pairs of chromosomes, 22 are identical in males and females, except one pair, which is also known as sex chromosome, which is XX in females and XY in males. During fertilization, if X of male fuses with X of female, female child is born. If Y fuses with X of female, then male child is born. Mutation. Mutation is a phenomenon which results in alteration of DNA sequence, which ultimately results in the change in genotype and phenotype of an organism. Mutations are caused by mutagens. There are two kinds of mutations, that is chemical and physical. Pedigree analysis. An analysis of traits in several of generation of a family is called pedigree analysis. When you see how the pattern is followed, this can be used to detect any disorders which were inherited in a family generation after generation. As it's said, if diabetes is present as a family history from the maternal side, then there is a higher risk of all the female children of every generation to become the carriers of diabetes, and then their children to be carrying the same kind of disorder. If you have a closer look here, it is clearly explained that these are affected males and these are affected females. There are several genetic orders which are caused by mutation. Genetic disorders are of two types, Mendelian and chromosomal. Mendelian disorders are haemophilia, sickle cell anemia, phenylketonuria, and haemophilia. Sickle cell anemia. For each pair of genes, each parent passes on one pair to the child, and then the mother has sickle cell trait and the father with sickle cell trait. So when they cross, then there is one normal child and then two children with sickle cell trait, and one definitely has a sickle cell disease. In PKU, the phenylalanine changes to tyrosine. Sex chromosome disorders. These are the abnormal chromosomal combinations which are found in different diseases. Please have a look. Thank you so much. I hope you understood everything well enough. Please don't forget to comment below.